Three international don't do this. High repute. Dr. Bharat is also a recipient of the Young Scientist Award from the Vellore Institute of Technology, Vellore, for his research in the area of systems biology. He is currently leading the computational biology team at Atrimed. This team is involved in the development of plant molecules, database, and screening of the plant small molecules for desired activity using molecular modeling and simulations. Welcome to the session, sir.
Now I would like to invite Dr. Rishikesh Damleji sir, CEO MD Atrimed, to chair this session.
Thank you very much to organizers. It's my pleasure to be here. Respected co-chair, Dr. Manoj Kalur, and respected scientists, Dr. Vaman Rao, Dr. Shivashankar Balasubramaniam, Dr. Lata Damle, and Dr. Ramji. I suspect whether Dr. Ramji is already there, or I think we have to. I think he's yet to join. Somebody can contact him. Now, see, there are different ways of looking at one subject. It could be that we look at a subject as vaguely as diet, or as vaguely as in a holistic manner, where it is just for trivial diseases or preventive, or some kind of relaxed manner at which you approach a disease. Whereas some of the serious disorders, even if they are chronic, need to be addressed very rigorously, including rheumatoid arthritis, psoriasis, or even the chronic diseases, even diabetes mellitus or hypertension. Though these are silent killers, they may not kill you acutely. They do require appropriate intervention. And when I say appropriate intervention, it needs to be very precise. And if you have to look at precise intervention, you may have to do a lot of research, and that research needs to be backed by the latest technologies. It wouldn't work with kind of a holism talk. So we have here some distinguished scientists who have done very peculiar research. Dr. Vaman Rao has worked, and he's working on how to neutralize snake venom, viper venom, as well as cobra venom. We will be interested to listen to him, and he has got remarkable results. Dr. Shiv is my friend, and Dr. Shiv is amazing because his in-depth knowledge of immunology is very exciting. And when we talk about immunology and immunity, sometimes we are very complacent. We say that immunity is good, we'll improve your immunity, etc. But immunity also has got good components, bad components. You already know that IL-6 cytokine storm with exacerbated IL-6 could lead to disaster in COVID. So he's going to talk to you a lot and give you a gyan about what is good immunity, what is bad immunity. Is it good to improve immunity, or is it sometimes are there various ways of controlling the immunity? Thirdly, we have Dr. Lata Damle, who is one of the very few Ayurvedic doctors who understand chemistry. She has taken pain to understand phytochemistry, then later on computational chemistry or computational biology. From there to synthetic chemistry. Sometimes you might be wondering, why do you have to really look at synthetic chemistry when you are talking about Ayurveda? Just imagine that you are going to work on a chemical which is from an endangered plant, and all you can do is synthesize the molecule in the lab and start testing it. And once you test it and test positive, then think, how do I go about it? Whether I go about growing the plant again, or do I go about synthesizing the molecule? So even synthetic chemistry is also very, very important, as well as medicinal chemistry, where PKPD and absorption, distribution, and things are concerned. So she's going to talk about it. And lastly, we have Ramji. Ramji is a very distinguished professor at Cardiff University Department of Health Sciences. He is going to talk to us about, he has done a lot of work with phytomolecules and with using latest technologies. So he'll be talking about it. Dr. Manoj Kalur, if you want to say a few words, yes. Otherwise, you can sum up in the end. Already, our time has been trimmed off. We have short of 45 minutes. If it is okay with you, with your permission, I will ask Dr. Vaman Rao to start his discourse. Please. Yes, sir. Dr. Vaman Rao, please go ahead, sir.
Yes, sir. Is my slide visible?
Not at this moment. Yeah, now they are visible. Can you make it full screen?
Yes. Is it visible now?
Yes, sir. So I'll mute. Please go ahead.
Yes, sir. Thank you very much. I would like to express my deep gratitude and thanks to GAF. At the same time, I would like to also thank Dr. Damle for nominating me to give a lecture in this plenary session on this topic, which is very close to my heart. And this is a traditional medicine which used to be practiced by my ancestors. And we were living in a village where snake bite victims used to be there once in a month at least, because it is surrounded by estates. And the primary health center did not have any medication required for these snake bite victims. And they were dependent on our family. And this medicine has been, in fact, traditionally practiced in our family, which I'll be talking about it in few minutes. And because you know that the snake bite is a most neglected disease in the medical field. Why? Because it has become very common. If somebody gets bitten by snake and dies, and sometimes the treatment is available, medical facility is available nearby, and sometimes the medical facility is not available. But it is very, very devastating for a family which is very poor and dependent on a bread earner who passes away because of the snake bite. Sometimes they don't respond even to the traditional medicine that is available, that is especially the horse antivenom. And this is the statistics that I've got recently from the Hindu newspaper. Every year there will be about at least 35 to 50,000 mortalities because of the snake bite in different parts of our country, and about 140,000 disabilities which are associated with the comorbidities of the snake bite. And the prominent snakes, all of us know, they are called as D4, cobra, Brussels viper, crate, and saw scale viper. And whatever the medicine that has been practiced till date, some traditional medicines are used, and they have saved the lives of people. And wherever the medical assistance is available, the horse antivenom has been administered. But there are many complications associated with the horse antivenom. One, it needs the medical assistance, the medical practitioner or a nurse to administer it intravenously. Second, it needs to be stored in a refrigerated facility. Otherwise, it loses its activity. It undergoes denaturation. And many remote places in India, villages, where there is no primary health center available, plus even the electricity supply may not be there, and refrigeration facility not available, the storing of horse antivenom becomes very difficult. So therefore, such victims who are in the remote areas, working in the remote fields, sometimes when they get bitten, it is too late for the medical assistance for them. And before they reach the medical center, they might pass away. Then the horse antivenom, whenever it is administered, everyone knows that it does cause anaphylactic reactions and other comorbidities. Similarly, at the same time, sometimes the horse antivenom is in short supply. It is not available because it is not supplied to that center. So that becomes a very difficult situation for the victim. And the medical center may not be there in nearby area. So therefore, another most important aspect is about the cost of the horse antivenom. It is very costly and a poor man cannot afford it. Therefore, the Ayurvedic treatment through the herbal remedies assumes significance, at least in the form of first aid to save the life of the person. There are many, many documents available in the literature, and most of them have worked, and many scientists have worked in India on different types of the herbal remedies on the snake venom, carried out research on the snake venom of different species of the snakes available. The predominant of them are just listed, but it doesn't mean that this is the reference that are available. There are many more references available. One of the prominent one is Andrographis paniculata. A lot of work has been done on this plant to study it as an antivenom property on Naja, Daboyora venom, and other that is saw scale viper venom. Similarly, the hydrocotyl javanica and gloriosa superba. So then Pentase burmanica is another plant which has been worked on Naja Kautia venom and prevents lethality. Hemidesmus indicus, also known to prevent lethality and hemorrhagic activity of viper. All these are experimental evidences on the animal studies and not on human studies. And maybe some tribals might have been using these medicines and they have been practicing it to treat the victims of the snake bite, and it has worked in the traditional medicine in the tribal belt, tribal areas. So going to my topic on which I have worked, the plant material, that is Coax lacryma jovi, the root extract. This was our family medicine, and this used to be given to the snake bite victim by my ancestors, and after the death of my mother, it stopped because we are all urbanized now. We don't practice this Ayurvedic medicine, but it remained very close to my heart to carry out some research on this. And one of my PhD student, Rajesh, worked on this, and we did find some good results as far as some of the enzyme activity inhibition, in vitro enzyme activity inhibitions are concerned, like hyaluronidase and L-amino oxidase. And these are the papers that we have published from the work carried out using Coax lacryma jovi ethanolic root extract. And we found that the ethanolic root extract is not toxic in the rat or mice at the dose of 2 grams per kg. And we also carried out some necrosis and hemorrhagic anti-hemorrhagic activity with the root extract at a dose-dependent manner, that is 100 milligrams per kg, 200 milligrams, and 400 milligrams per kg. And you may see to the extreme left of yours the control where the animal skin at the dermal injection was given. 30 microgram of Dabaya Russell venom was given, and at the same time it was treated with the standard horse antivenom. And you may see the decrease in the size of that hemorrhage only a little bit, but with the treatment of the ethanolic extract, which was administered, okay, orally to the animals, showed significant results and decrease in hemorrhagic activity. And we did carry out some of the in vitro venom enzyme inhibition studies, and we did find good results with venom enzymes like hyaluronidase, phospholipase A, and protease. For hyaluronidase, we used hyaluronic acid, which is available commercially, and phospholipase A, we used human RBCs, which were washed in the physiological saline to find out the hemolysis of RBCs. And for protease, casein hydrolysis activity were carried out using standard procedures of these references. And all 4 snake venoms we tried for the anti-venom activity with the Coax lacryma jovi extracts. We used 4 different types of extracts. First, dried root ethanolic extract at 100 microgram and 200 microgram per kg level, and fresh root ethanolic extract, that is just fresh root taken from the plant and macerated in ethanol at 100% maceration, and 100 microliters and 200 microliters we used for the test. It is all in vitro experiments, not in the animal studies. I'll come to it a little later. And we also tried lyophilized aqueous extract, which is stabilized with 2% starch. And I'll come to the point why we had to stabilize with the 2% starch a little later. And the fresh root aqueous extract. And you may know that fresh root aqueous extract will be useful only at the site or very close to the place where the victim is bitten by the snake. But it is not stable and you cannot keep it in the laboratory temperature, the fresh root extracts for long duration. But only for the experimental purpose, we have used the fresh root extract. Now we have the results here. So 50 microgram of Naja venom, hyaluronidase activity was studied, and 100 microgram of hyaluronic acid was used. And you see here the results. In fact, the dose-dependent manner inhibition of the activity was observed, and the activity with dried root ethanolic extract was found to be higher than the fresh root ethanolic extract or the lyophilized fresh root extract or the fresh root extract. Similarly, we carried out work with WRSLE venom, and we found a similar activity up to 40% inhibition with the dose-dependent manner, and with the Echis carinatus venom, hyaluronidase activity, the dose-dependent manner inhibition was observed, and with Bungarus cerulus also. Then we carried out work with phospholipase activity, where human RBC, 100 microliters of human RBC was used, and 50 microgram of Naja venom was added. Before that, the root extract was added to it, and inhibitory activity was studied. And we measured the hemolysis of RBC using the UV visible spectrophotometer, and percentage inhibition was calculated. And you see the activity here. Quite good activity was found in all the 4 different types of extracts, with Naja venom, WRSLE venom, and Echis carinatus, and Bungarus cerulus. Then we carried out protease activity, where casein, 2% casein hydrolysis study was carried out, once again using the UV visible spectrophotometer, and percentage inhibition was found. And there was good activity with dried root ethanolic extract and fresh root alcoholic extract for Naja venom protease activity. WRSLE activity. The few differences are there. In some, the fresh root is showing good activity, and some dried root is showing activity. But most important point is there is inhibition activity in the root extract. This is Echis carinata protease activity inhibition, and Bungarus cerulus. Then we did carry out the fibrinolytic, that is inhibition of fibrinolytic per kg ethanolic extract for the WRSLE venom as well as Naja venom, and there was significant inhibition. I have not projected the graph. Similarly, 100 microgram ethanolic extract was also capable of showing nuclease inhibition activity of Naja venom. And some activities carried out in animals where with the WRSLE venom, where hemorrhagic activity was neutralized with 400 microgram of ethanolic extract, and 30 microgram of the WRSLE venom carried necrotic activity was inhibited by 400 microgram of ethanolic extract. Then 2 microgram per kg WRSLE venom and 4 microgram per kg Naja venom edema was inhibited by 400 microgram per kg ethanolic extract. Then coming to the actual in vivo studies, that is with the mice, we carried out a lot of other preliminary studies of LD100 dose for different venoms, but we could not do the work with other species because of lack of funds. We have done part of the work with WRSLE venom, LD100 dose for the intradermal, LD100 dose for subcutaneous, and LD100 for intramuscular. Here is the dose that is mentioned, 8.5 microgram per gram venom of LD100 for intradermal, and 10 ml per kg aqueous fresh root extract was given in 3 doses at hourly interval, and 6 animals, there was no mortality. Similarly, subcutaneous LD100 dose was 9.8 microgram per gram, and fresh root extract was 10 ml per kg, and 3 doses hourly interval, no mortality. Intramuscular, 8.5 microgram venom, and similarly, there was no mortality. Then coming to the other type of extract, here we treated the animals with the LD100 dose, that is injection, intradermal, subcutaneous, and intramuscular, and fresh root extract, aqueous ethanol extract. Fresh root ethanolic extract was administered, and it was diluted 1:5, and the extract was 50%. That is 50%. That is 50 grams of ethanol. 50 grams of root was macerated in the 100 ml of ethanol, and that was diluted 1:5 and given orally. And see, 4 animals were treated in each group, that is intradermal, subcutaneous, and intramuscular at LD100 dose, and there was no mortality. Then the lyophilized extract. Why we had to carry out this work? Because I wanted to see if the root extract can be lyophilized and used as a stored material. And if it could be transported to the remote areas where it can be kept in laboratory temperature and utilized by a person as a first aid measure. I wanted to do the study. Therefore, I prepared a lyophilized fresh fruit extract. When we first prepared plain lyophilized fresh fruit extract, we did not get good protection. There was more than 50% mortality. Then one of my scientist colleague from Pennsylvania University told me that some of the active ingredients of the root extract may get denatured when you lyophilize it. It is better you use dextran or starch or any other stabilizing material, which tricosan or something like that, which will stabilize the material. So I use simple starch, 2%, and lyophilized root extract was used, fresh root extract, which is aqueous. And we found with the LD100 dose, when 500 milligram per kg, it is powder, which is given at a dose of 500 milligram per kg, and 3 doses were given orally, and it was found that there was 100% protection against the tuberculosis. So my contention is this preparation of the coax lacrymal jovi, if it could be used as a first aid measure to prevent the death of the individuals, it would be worthwhile. Here, I'm talking about stability of the extract. We found that the ethanolic root extract, fresh root extract, prepared and kept in the laboratory temperature for 1 year showed the same effect, same activity, and 100% protection in the WRSLE venom and LD100 dose. Same way, lyophilized root extract kept in the laboratory temperature for 1 year also showed 100% protection against WRSLE venom. So my contention is there is no need of fridge or there is no need of freezing facility. The root extract in the ethanol or in the lyophilized form is stable even after 1 year, and it could be used as a first aid measure for the persons who are bitten by the poisonous snake. It could be possible that we have to carry out some more work with the Naja and Echis carinata, Bungarus cerulus. Possibly we may get same type of results. Due to lack of funds, I could not carry out much of the work in these other species. So we have seen that there is 100% protection against WRSLE venom at LD100 dose intramuscular injection of WRSLE venom to the mice, as well as intradermal or subcutaneous. And I still feel that it can be used as a first aid measure. And I would like to give my acknowledgement and thanks to my PhD student, Rajesh, who has worked extensively on this topic. And I would like to thank Dr. Murali, Deputy Director, New Care, Nite University, who helped me with the animal experimental studies. I also would like to thank Dr. Rishikesh Damle, who helped me and encouraged me to move forward with this work. And he's my, in fact, I should say that my close partner. And I would like to come out with my product with him. And I would like to thank Birak Pace for giving me partial grant, and only the first installment grant they have given me. And I would like to thank Irula Snake Catchers Cooperative Society for giving me the venom. Thank you very much. If there are questions, I would like to answer those. Thank you.
Can you guys hear me?
Yes, sir. Yes.
Yeah. Thank you very much for that enlightening work, and I've seen through this work you are doing since many years, and I know that you have also done further work on purified molecules from these plants. Anyway, probably you have your own reservations on sharing the data. That's fine. I want to ask you first question. Meanwhile, subsequently others can ask their questions. First question is, what was your idea when you went on doing these enzymatic assays?
Yes, sir. First of all, we wanted to see whether actually this works on enzymes of the snake venom. Those are the major toxins that are found in the snake venom. And in vitro, if we can prove that these are getting inhibited, then we can carry out the work with in vivo studies And possibly that must be the mechanism which will work in human beings also, where the enzymes of the snake venom are getting inhibited when this preparation is administered orally to the victims.
Okay, thank you very much. Welcome, Ramji. I see him there now.
Hi. Thank you.
Welcome. Yeah, to continue with Vaman Raoji, I think you also had a group of animals as control, and
yes.
which was not shared. I think you should have shared that. How many animals died in control group?
Control 100% mortality, sir. That's
Within
right.
how many hours?
Within 2 hours.
So control group 100% mortality within 2 hours, and
Yes.
others did not die. Basically means what? In the end, you had to sacrifice them.
Yes, because
Okay.
it is not advisable to keep them.
Yeah, and I understand.
Somebody else uses it for some other experiment, it will be
that-
bad for him. Yes.
Yeah. I just get it. So any other questions you can ask, including organizers? Dr. Manoj?
Hello.
Yeah. Any question?
Yes, sir.
I do have a question. The thing is that how far is it to make it available for each human? The thing is that for that trial, what has to be done?
I need help from the doctors. I need clearance from the ethical committee. Then only we can prove it that it works in the humans. But in my village, the primary health center doctor himself used to send the victims to our house. Why? Because there was no fridge available in the village. Plus, there was no electricity at that time. I'm talking about 1960s to 1980. And the horse antivenom was also not available. If at all, we had to send the victims, we had to send about to 32 kilometers away, which is Chikmagalur district from our village. And you know that at that time, bus transport was also not available, and vehicle transport also was very poor. Our village connectivity was also very poor. And the doctor himself used to send the patients to our house, go to that house, they will give the medicine, and the victims used to survive. And that was my childhood observation. And that, in fact, kindled interest in me that as I became a scientist, biochemist, I should investigate and find out what is actually working. So I started my work, and I'm continuing my work.
Yeah, thank you very much. I think there is a requirement of political will as well. The large scale, preventive field studies can be done in diseases where snake venom is endemic, and to see whether it can be given as a first aid at the point where you're bitten by the snake, because you can store it right in advance and see whether the mortality rate goes down. So anyway, we can talk about it. Thank you very much, sir. It was a good presentation. Later on, the vice chair will sum up some of the points what you have put forward. May I now call upon Dr. Shivashankar Subramaniam to deliver your lecture, please.
I need to be given the permission to share my slides. Can you all see?
Yes, sir. Yes.
Yeah. Thank you. Thanks a lot for for giving this opportunity, particularly the organizers for this meeting and Dr. Damle. It's good to see some familiar faces. Professor Deepak Ramji is there, our cardiac connection, and it's nice to meet always people who you know from your long time. So yeah, it's a pleasure to be here. So the topic that I've chosen today for is immune modulators, good or bad during pandemic. And the reason for that I chose this topic is, this is a data that is coming from Google Trends. You can see that how the term immunity booster has spiked since the pandemic has hit, and you can see that India is the epic center for that search engines, for the keyword search. Everybody was looking for how can I increase my immunity so that I don't get the COVID or Corona. That's the logic behind it, and which is natural. Like I don't think it is anything wrong in doing this search, because everybody wants to safeguard themselves and their families, loved ones, and so on. So they thought, is there any way, when the world had no clue how to protect themselves, is there any way we can protect myself by increasing the immunity? The good thing is they all knew that, if they have immunity, if they increase the immunity, they can get protected. That's half the education is already done there, I think, for the general public. So I think this really prompted me to look into, what is this? Why people are so, curious about this, and supporting this is a lot of products that came into the market in the last 1 year. I can say that, almost every day you will see a lot of things. I'm sure all of you must have seen these sort of things in the news and in the newspaper and various social media, saying that this does this and so on. So I thought maybe we need to look into it. Is this really good to take an immunity booster during pandemic? And that prompted the title for this talk. So, just to give you, the questions that we want to address here is, do these products really modulate immunity? Whether we have any answers to this, let's find out. And is it wise to modulate immunity during COVID pandemic? And that's the sort of questions that we will try to address. And in order to address this, we need to ask these questions. What is immunity? And given that this is a fairly different set of audience who's not specialized in immunology, I thought it would be good to cover some of these basic aspects, and also ask the question whether can immunity be modulated, and what really happens during COVID in terms of immune response, and then the pros and cons of modulating the immune response during COVID, and can nutraceuticals or phytoproducts modulate immune response? So these are the questions that we will try to address in the next 30 minutes or so. Do I have 30 minutes? I just want to check.
Yes, you have 30 minutes.
Thank you. So what is immunity? I think, again, there are different sets of definitions. This is something which I found it to be appropriate. Immunity can be defined as a complex biological system endowed with the capacity to recognize and tolerate whatever belongs to the self, and to recognize and reject what is foreign, which is non-self. This is again a textbook's definition of immunity. And what really we are talking when we talk about immunity is it's a complex system of cells. Like there are two arms. One is called the innate immunity, one is called the adaptive immunity, and you can see that and they also bridge. There are two arms. One is the innate and the adaptive immunity, and then within the adaptive immunity there are different lineage of cells that comes and plays a role at different stages. I'm not going into the details, but all I want to know for you to draw your attention is to these CD4 cells, CD8 T cells, and the B cells. These are the ones that play a critical role during viral infections, and all vaccines that we are currently aiming to take or trying to develop is all towards these cells. So this is something which we need to keep in mind, so that I will come into when we talk about whether it is worth having these immunity boosters, good or bad. We will go into that. Okay. So again, cell-mediated immunity, which is your the two components that I talked about, which is your T cells and B cells. The first thing that we need to understand is both of them requires an exposure to the antigen. That is, means unless you get an antigen exposed, you can't have immunity, whether it is B cells or T cells. And basically, what happens is, this is how the mechanism works. The antigen presenting cell takes the antigen, and then it presents it to the T cells, and then it elicits a B cell response, which then goes on to produce antibodies. And that is what we are currently measuring in COVID. Positive people, whether they have got IgG, then you can say that they have got exposed to the viral infection, and then they have got an immunity against it. On the other side, we have T cells. Again, similar process. The T cells are, the antigen presenting cells takes up the viral antigen, and it goes and presents it, and then the naive T cells, which carries the T cell receptor for the viral peptides, gets activated, and then they go on to differentiate, and then they further differentiate into T helper cells, cytotoxic T cells, and then that's how they go and kill the virally infected cells. These are the two, and both these mechanisms requires prior antigen. And without the prior antigen, the question that we need to ask when we are trying to take immunity boosters is, where is this immunity coming from for your body? Okay. So again, acquired immunity, this is called the acquired immunity, which is lifelong, and when you have this acquired immunity through the antigen exposure, it causes an immunological memory, and it protects you throughout your life. And it can be an active immunity, which you get it through an infection, or a passive immunity where you get this through vaccination or through the mother's feed and so on. So this is a short term, but it gives you still protection. And this is where the convalescent serum therapy or the plasma therapy comes in, where antibodies from an infected person will help you to recover from an infection. So which is a short-lived support system. So the point that I want to make is, immunity is not something on a linear scale. You over a period of time, I take today something and then the next day something, your immunity is going to increase. It doesn't work like that. Rather, immunity is a complex system. It is a fine balance between the various parameters. When it exceeds that, you end up in getting an autoimmune diseases or allergic sort of conditions. When it falls below a threshold, it causes you infection and cancer. So in summary, I can say that immune functions vary between subjects and fluctuate over time within a subject. In healthy population, these fluctuations define the boundaries of a normal range and immune functions, providing optimal health. And genetic makeup, past experience contribute to the differences between the subjects, and of course environmental, lifestyle, all this play a role in how your immunity or the immune system works in your body. Again, as I said, it is not a linear scale. It's rather a fine balance between various parameters so that your immune system is at optimal health. It doesn't go above a certain level to cause autoimmune diseases, or it goes below a certain level where you become more susceptible to infection and cancer and other conditions. So that's a sort of overview, about how the immunity works. So can immunity be modulated? That's the question then. Can this be modulated in any way? Yes, there are instances where immunity has been shown to be modulated, because some of the parameters by which we measure immunity is the cytokines that play an important role in augmenting these immune responses. And this is an example of an animal study where you can see an animal which is left freely to move, which is the no stress condition. There is no increase in the TNF alpha levels in their circulation. Whereas when you put the same animal into a bathtub and then you made them to swim, the stress causes an increase in the cytokine levels. And then when you restrain them in their hands, again it increases the cytokine level. So this really tells you even stress can modulate your immunity. So it is important to keep that in mind. That when we are talking about Ayurveda or holistic health, not to get stressed and do things that will take away, de-stress you. So again, IL-2 not as prominent as TNF alpha, but very similar trends you can see. And this is yoga also shown to be influencing immunity, and you can see that the gray line is people who are highly practicing in the yoga. The blue line is people who are low practice frequency, and the yellow line is the no practice at all. And at some point, they all had similar levels of cytokines. And as they started practicing yoga, you can see that very similar trend between IL-6, TNF alpha, IL-1 beta. They are all pro-inflammatory cytokines. You can see that it goes down. It is not like when you do yoga, these cytokines doesn't go up, rather it goes down. So it is, what it means is just because you have high level of cytokines does not mean that you have heightened immunity, rather you have optimal immunity. So that's what we need to note here. And again, this is another conditions. This is again in the allopathic medicine. We can see that cancer is being targeted by looking at targets that are known to play a critical role in in organizing the immune response. So these are all various targets that are currently, I'm just citing this as an example that immunity can be modulated, but you need to have good scientific understanding about how this whole immune system works. I'm not going into the details of this slide in the interest of time. And this is again another condition, which is autoimmune diseases, and there are several targets that are currently being explored. The cartoon on the left-hand side talks about the various targets that are currently in clinical trials targeting various immune modulators or immune mediators, and they are all in current clinical trials. So the right-hand side, you see a product. In fact, this is from Atrimed Pharma, and this is currently used for psoriasis. And I'm not going into the details of how this works, but all they know I can tell is that it blocks IL-17, which is a key mediator of inflammation in psoriatic conditions. So again, these sort of bringing the understanding the Ayurveda, how it works through the modern day science is the future, I believe, and this is something which I think will evolve more in the coming years. More and more information probably will emerge from the Ayurvedic approaches from a scientific point of view.